Hello everyone and welcome to the presentation on prions and viroids. Just to inform you, in the last few videos we were talking about viruses, how do we describe them using cryptograms, and how do they replicate. If you don't know about these topics or subjects, you can refer to them. You will find link to the previous videos over here somewhere on your screen. Alright, now let us jump into prions and viroids. So prions or viroids are also infectious particles like viruses, but they are more primitive than viruses. Why am I saying that they are more primitive than viruses? Let us see here. Alright, so viroids are actually smallest known infectious agent till now. Smallest known infectious agent, and they are only a nucleic acid entity, only a nucleic acid, and mostly RNA. So nucleic acid entity. So what do I mean by this? Let us try to understand this point. Okay, so far we have studied that viruses consist of a capsid layer like that, and they have a nucleic acid inside them. Okay, they may be DNA or RNA, and they also sometimes have an envelope surrounding them. Okay, so far so good. What are these viroids, and why am I saying they are more primitive than viruses? Let us see. Viroids are actually, no protein is there in viruses like the and no protein is there on viroids like that of the viruses, the capsid layers of viruses. They are just a nucleic acid, and this nucleic acid is a coiled RNA. Just that. So how do they infect other organisms? Just like the mechanism of the virus, if they enter the cell, they can control the machinery of the cell, and they can the uh, destroy the cell's DNA or genetic material, and then replicate itself using the machinery of the cell. If you want to know more about the mechanism, you can refer to the previous videos. Alright, so this was about viroids. Let me give you some more information about the viroids and the diseases that they cause. So let me put it aside and let me continue my text box. So diseases caused, let me enlarge the text box a little bit, caused by viroids are: first of all, it is the potato spindle tuber disease. Alright, so that is a disease caused by virus, and as you can see, the name suggests it is caused in the potato plant. So potato spindle tuber disease. So that is one of the disease that is caused in potato plants. I will not go into detail of this disease because it is out of the scope of this video. Now, some more diseases are chrysanthemum chlorotic mottle stunt. If you want to know all the details about this video, you can do a Google search. You will find a lot of details. So let me write the disease: chrysanthemum. And if you know what is a chrysanthemum, it is also a kind of plant. Chrysanthemum chlorotic mottle stunt. Chlorotic mottle stunt. Okay, another disease, and there are many such disease. like this, and these viroids were discovered first by Diener and Raymer. Discovered by Diener, I'm sure I'm mispronouncing it, Diener and Raymer in the year 1971. 1971. So not very old. Quite a new discovery. So that's all about the viroids. Not much. They are the same as that of the viruses. They lack only the protein coat or the envelope outside them. Okay, but the prions are a lot more interesting. So let us start the prions down below over there. Okay, so what are the prions? Well, as I described, the viroids are the infectious nucleic acid entities. Prions are infectious protein entities. So prions are infectious. So let me enlarge the text box. That much will do it. Infectious protein entities. Alright, so prions are infectious protein entities, but this sounds quite confusing, because how can proteins be infectious? This is quite confusing, right? Because how can protein infect a cell? We know that DNA or the nucleic acid can infect a cell. They can enter a cell and they can destroy the genetic material of the cell, gain control of the cell and all that. But how can a protein infect a cell? And you will be more surprised to know that these prion proteins are the proteins of our own body. They are proteins present in our own body, but they are misfolded. So let me say all the things in a bit more detail. So for that I need to draw a neuron first. So the diagram will be not very good or very accurate. I am drawing it very fast, so don't just laugh out with my diagram. Okay, but this neuron I am drawing just to explain you about this prion proteins. So this neuron will have a cell body, and it will have the axon terminals like that. Not very good. I am drawing it in paint using my mouse, so more difficult to draw. So but this will do the work. Okay, so as you know, these neurons have the dendrons. These are the dendron portions, and these are the axon terminals, and another neuron can be connected to these axon terminals by synapses. Yes, so another neuron can be connected to these axon terminals by synapses. So whatever it is, like that. So why I am telling all this? This synapses connection has some proteins involved in it, and here the prion proteins come into action. The prion proteins help the neurons to connect with each other. So in this synapse there is the prion protein. Here somewhere the prion protein is there in everyone. Alright, so prion protein, and they are the normal prion proteins. Protein. Alright, till now well and good. These prion proteins do not affect, or in other words, they help us. How do they help us? They create the connection between neurons, and as if you know that if the neurons are connected, and they create the permanent most strong connections between the neurons, which helps us to retain our memory and to recollect some past incidences, to do all that. So these prion proteins actually help our brain with our memory. So they are very useful. Then how do they turn into such a disaster? Let us see. Sometimes, and also the prion proteins look like this. Alright, so let us see how they look. They are the alpha helices like this. They are the alpha helices structure. They look like this, and as you know that the alpha helices are present, the proteins which are alpha helically coiled are present in the cell membrane, but due to some radiation or some it is very nobody knows how this is happening, but due to some factor this alpha helices are misfolded and they become into beta pleated sheets. If you know, will be very confusing if you don't know all these protein foldings. I will make some more video on this, but this alpha helices will convert into the beta pleated sheets, and this beta pleated sheets are not made for to be present in the cell membranes, so they cannot be present here anymore, so they have to move out of this. So when this beta pleated sheets are formed, this stage is called the prion protein. Alright, so here now it has become an infectious prion protein. So let me write down over there. This is a normal prion protein and we represent it by PRP. So we represent it by PRP, and we represent the infectious prion protein by PRP, and in the subscript, don't know how to make the subscript, let me do like this, C, like that we put. Alright, so it is a PRPC, a complement, and this beta pleated sheet will have the same amino acid sequences as that of the alpha helices. They are just folded in a different way. Now how do they affect it? So once these prion proteins are moving out of these synapses, now the neuron junctions cannot form, the synapses cannot form, because they are the ones responsible for connecting neurons. Once they are not there, the neurons cannot connect together. So if the neurons cannot connect together, it will be difficult to send a nerve impulse through for our brain. So if the prion proteins are not there, the synapses will not form and the neurons will be not connected. And also once prion protein like this, beta pleated prion protein is formed, it is protease resistant. So what is this protease? Protease is an enzyme used to digest the proteins, but this is such a protein which is resistant to this enzyme. So this is protease resistant. This is a protease resistance protein. Okay, and how do this replicate themselves? So like the viruses replicate themselves by gaining entry into the host cell and then capturing the cell machinery, they replicate themselves by encouraging other PRP proteins to fold like them. So if there is a PRP protein, let's say here, this is a bad protein, they will go to other neuron synapses, and then they will interact with the other good proteins. So let me do the good proteins with green. So there will be other good proteins. Let me make some over here. Okay, so there will be other good proteins like that over here in the neuron synapses, and there will be also here connecting to another neuron which can be over there somewhere. So this prion protein will interact with this normal PRP protein and make them into another prion protein. So it will again become a prion protein, and these proteins will again become a prion protein. They will again go to other parts of the nervous system. They will again interact with the good proteins and make them again into bad prion proteins. So that's how they make more of themselves, and that's how they infect us. What is the bad part? How do we get infected by this? When the synapses are not there and this nerve cell becomes useless, right? If the nerve cell is not able to conduct the impulse through them, they are actually useless. So what happens is there is special cells called the microglial cells. So there will be a special cells, they are phagocytic cells, and they are known as the microglial cells. So they are known as the microglial cells, and what they do is they just engulf all those cells which are useless, and they digest them and they remove them. Alright, so they will come over here, they will eat up this neuron and all other neurons which are useless, and they will just digest them. So at last we will find, if we do a if we see the brain of a prion protein infected individual, we will find like this. If this is the brain, let's say this is the brain, here's a very rough diagram, just quickly drawing. Then there will be many holes in the brain like that. Why are the holes formed? Or let me choose another color. Let me choose the blue color again. There will be many holes in the brain. Why are the holes formed? Because here the nerve cells have become useless and they have been digested by the microglial cells. So this brain looks very much like a sponge. Sponge contains many holes with air in it. Like that, this brain has many holes, and such a infected prion protein infected individual are not able to coordinate their body. They are not able to send nerve impulses to various parts of their body because their nervous system is damaged, and such individuals are really paralyzed or mad like this. So we have discussed in a lot detail about the prion proteins. So let me write some more things about the prion proteins. Okay, so I have already mentioned that they are infectious protein particles, and they were discovered by Alperath et al. In 1966. Alperath et al. in, sorry, let me write down discovered by, or else you will not understand what is Alperath et al. Okay, discovered by, and let me enlarge the text box as always. Discovered by Alperath et al. in 1966. Okay, and infectious proteins. And what are the diseases caused by these prion proteins? Some diseases caused by prions are the mad cow disease, or the scientific name for the mad cow disease is bovine spongiform encephalopathy. Alright, so mad cow disease, and as you can see from the name, they are caused in cows. There is scrapie, okay, and there is also diseases like Kuru, which are caused in human beings. So Kuru is also known as laughing death in women of tribes of New Guinea. So they were first found in the women of tribes of New Guinea. So just for your information, I mentioned that. So there are some diseases, and that was about the prion proteins. I want to mention some more terms about such more primitive infectious subviral agents. Some terms is the virion. You might come across some terms called a term called virion. And what is the virion? It is basically if a virus and causes its DNA material or the nucleic acid to it puts the nucleic acid inside the cell, then what is remaining? The capsid and the lipid layer, if it has any, that part is called the virion, nothing else. And you may also here the term virusoids and anything, the pronunciation is not much important, and these are actually viroids linked with larger RNA of true viruses. Alright, and they replicate within their host and they do not cause any infection, and they were discovered by Randalls for your information. Alright, so I hope you didn't find this in video too much confusing, or if you find it, I apologize for it, but you will find all these protein foldings and all these things. You will learn about this more if you study the chapter biomolecules or wait for my videos to come. And okay, we will meet the next time. Bye for now. Thanks.